This program is brought to you by Emory University.
I am very pleased to introduce our speaker today, who studies the topic actually very close to my heart. Looking forward to talking to you later. Dr. Nadia Sutton is an assistant professor in the Division of Cardiovascular Medicine at Vanderbilt University. She received her undergraduate degree at Loyola and then went on to earn an MPH at Boston University and then a medical degree at Loyola University. She then went on and completed her internal medicine residency at the University of Michigan, cardiovascular fellowship at the University of Michigan, including a year as chief fellow in interventional cardiology at the University of Michigan. She then joined came south to the better weather, I assume, to Vanderbilt, where she is currently an assistant professor. Looking through her CV, she's really done an excellent job combining research with her clinical duties. She has published some impressive publications, including a cover story in ATVB on CD73, which I think we'll be hearing some about today. She's received some very interesting grants, including scoring in the top percentile for the American Heart Association Career Development Award, which is very impressive. So Dr. Sutton, looking forward to hearing about your work.
All right, thank you so much. Can everybody hear me okay? Okay, great. Well, thank you again very much for the invitation to give grand rounds at Emory today. I've, in my relatively short career so far, have had several colleagues that either trained or worked at Emory at various points, and I've just really enjoyed working with all of them, and I highly respect your program and look forward to hopefully visiting in person at some point. So today I wanted to talk a bit about ectonucleotidases and the scientific value of studying challenging problems. And maybe, the story that I'm going to tell you will be maybe more helpful for some of the individuals in the audience who are like maybe at my career stage or getting to that career stage, because my guess is that some of the more senior individuals in the audience probably have experienced some of these same pitfalls, which I'll discuss, and learn some of these same lessons along the way. And I know I've certainly benefited from hearing from the experience of those who are more senior than me, and learning from their scientific successes and failures. So here are my disclosures. And I had some stated learning objectives, so I'll just mention them. One is just to help the audience understand what ectonucleotidases are. I've been studying them for actually shockingly over a decade now, but I've sort of learned to respect them and try to understand them. And so, some of what I'll be talking about is, the actual mechanisms by which they may impact cardiovascular disease, and then just to generally explain some of the scientific challenges that I've encountered, and some reflections on my experiences as I've kind of gone through the physician scientist pathway. So first, I just wanted to start with what is an ectonucleotidase. So it's sort of a family of, I guess you could call them transmembrane enzymes. Some of them have sort of more than one transmembrane domain. So they mostly act on the surface of cells that either could be circulating cells like not neutrophils, lymphocytes, mononuclear cells, or potentially on the endothelium. They've actually been described as occurring in many different tissue types, although even in organs like the heart, they tend to be primarily expressed on the endothelial cells and on resident leukocytes, more so than on, for example, vascular smooth muscle cells or myocardial cells, although they, again, have been described as actually acting in those types of tissues. Basically, what they do is they catabolize extracellular purines, such as ATP and ADP. And from ADP, you could actually create ADP. or you could actually cleave off pyrophosphate and develop AMP. But in any case, they kind of work in sequence. And so the next enzyme in sequence, the next ectonucleotidase in sequence is after CD39 is CD73, which metabolizes extracellular AMP down to adenosine. So in some ways, you probably have some familiarity, or even if you hadn't ever heard of these before, with the idea of these circulating extracellular molecules, because we are familiar with medications like Plavix and other P2Y12 inhibitors, which essentially are preventing that circulating ADP from binding to platelets and causing additional thrombotic problems. But you can kind of imagine that these ectonucleotidases are almost essentially our natural homeostatic way of getting rid of these, what otherwise would be platelet aggregating and platelet activating extracellular molecules, and metabolizing them to things like adenosine, which we know reduces platelet activation and aggregation, increases vasodilation, and vascular smooth muscle cell relaxation. So in a nutshell, just to kind of summarize again, so these ectonucleotidases are found on the surface of our cells for the most part, and they're working to get rid of things in our bloodstream that could cause thrombosis and create things that could be actually vasodilatory and protective. And this is just another figure kind of demonstrating that pathway and where they're found and what happens to these circulating molecules when they're not catabolized in the extracellular space. So, and I'm just layering on a little bit more just to explain that even though I'll mostly be talking about CD39 and CD73, which are two of these ectonucleotidases that work in sequence today, they are not operating in a vacuum. So even though the enzyme kinetics can get a little bit confusing sometimes, and also when we're thinking about the molecules that they're metabolizing, it actually can be pretty tricky to try to capture the extracellular concentrations of these things like ADP and AMP and adenosine. And we all know adenosine is very short acting. That's why we can give it and it goes away within 30 seconds. In addition to that, there's actually additional ectonucleotidases, which I'm not going to talk extensively about today, but just because we'll talk about it a little bit later in the talk, I just wanted to point out this other one called EMPP1, which actually also catabolizes extracellular ATP to down to, instead of just removing the terminal 1-phosphate, actually we'll remove the terminal 2 phosphates and actually create pyrophosphate, whereas CD39 tends to act sort of in a more sequential manner and creating inorganic phosphate as its byproduct. And this is relevant because we do know that inorganic phosphate promotes mineralization, whereas pyrophosphate actually inhibits mineralization. And by mineralization, I'm referring to things like calcification. So for example, vascular calcification. So as you can see, and I hope I'm helping you to understand, this is a fairly complicated enzymatic cascade that happens in our bloodstream on a regular basis. And it's I consider it the equivalent to extracellular nucleotide whack-a-mole, because essentially these enzymes are kind of sitting around just waiting to have some ATP or ADP come by so that they can take care of that for us. So I also wanted to just mention that ectonucleotidases have been implicated in some human disease states. So besides the scientific interest in these molecules, they've actually been pretty extensively studied in the field of cancer biology, where cancer cells actually can sort of learn how to upregulate some of these ectonucleotidases. So for example, CD73, which is the ectonucleotidase that metabolizes extracellular AMP to adenosine. If that's presented on a cancer cell, that helps a cancer cell to essentially evade the immune system by producing more adenosine in its local environment and averting sort of an inflammatory response and being able to evade the immune system. Other areas where this has been extensively studied is in the area of thromboembolism. So my mentor at the University of Michigan, Dr. David Pinsky, had extensively studied ectonucleotidases in the field of stroke. and thrombosis in his work at Columbia and also at the University of Michigan. And also in the field of vascular calcification, there's a couple of diseases, ACDC, which is known as arterial calcification due to deficiency of CD73, has been described by Cynthia St. Hilaire, who's now working at the University of Pittsburgh in lower extremity vascular calcification, and GACI, or generalized arterial calcification of infancy, due to ENPP1 deficiency in about 70% of cases, which is a very rare genetic defect that would result in an infant developing vascular calcification. And then also there's been some implications in Hutchinson-Gilford progeria syndrome, which is another rare disease associated with premature aging. So I just wanted to start by kind of just letting you know about a little bit about my scientific background. I had worked when I was just out of college in a research lab at Boston University. The lab was the lab of Joe Scalzo, who is sort of a preeminent scientist, and I was lucky enough to land a job in his lab as a research tech, and then went on to continue doing basic science research while I was a medical student at Loyola, working with Dale Girding in a infectious disease lab. And so I really was interested in pursuing this physician scientist pathway. So when I came to the University of Michigan, I was interested in going into cardiology. And so I sought out my mentor, Dr. David Pinsky. And in that context, he had been interested in ectonucleotidases in the space of thrombosis. And so, we decided we were going to kind of look at a model of myocardial infarction. And I just wanted to put it in the context that around the time that I had started my residency, that would have been in 2008, there had just been these couple of papers published. You can see that they were published in 2007, demonstrating that CD73 and CD39 seem to be actually protective during cardiac ischemia reperfusion injury. So in animal models, there's different ways that you can actually cause myocardial injuries. You can actually essentially ligate the LAD and then allow it to be loosened after a period of time. That would be essentially ischemia reperfusion. Or you could actually permanently occlude the vessel, causing an infarct, which is essentially, it's a different sort of model of looking at different phases of myocardial injury. And so this was what was kind of known at the time when I was starting my residency. So this is a manuscript that I published. As you can see, it was 2017. So it actually took 10 years from the time that I was a resident to actually get this paper published. And it was a lot of work. And so, basically what we found, you can see there's an N46 to 48 per group. This is an murine model that, somewhat to our surprise, when we caused a permanent coronary occlusion, which basically means you put the mouse under a microscope and you ligate their LAD while they're on a ventilator. That surprisingly, the wild type animals actually did worse than the knockout animals. And we would have thought that the CD39, the presence of CD39 would actually be protective, and that the animals without CD39 would have done worse, based off of the prior literature suggesting that in an ischemia reperfusion model, that those animals, that the presence of having an ectonucleotidase on cells, and this was a global knockout, was actually protective in that model, whereas now we were seeing that it was actually harmful. And what we noticed was that the main reason that the animals died, which is not uncommon in the setting of a mouse permanent occlusion model, was that the mice actually developed cardiac rupture around day 4 after MI in the wound healing process. So the wild type animals developed the rupture, whereas the knockout animals seem to be protected. And part of the reason why it took us a long time to publish this paper was because it was counter to what was actually published previously in the field. It didn't make a whole lot of sense, and we were really kind of left with trying to understand why this would have happened. And we looked at so many different, possible mechanisms. Was it a TGF beta mechanism? So that's sort of a pro-fibrotic pathway. And because we did see actually more sort of fibrin in the animals that had no ectonucleotidase present, the lack of CD39. And there were some various other indicators that we were seeing sort of an improved fibrotic response to the severe injury. We also saw that in the animals without CD39 that they actually had more of an inflammatory cell infiltrate demonstrated here with flow cytometry and staining. And essentially what we came down to was that what we were seeing with this inflammatory infiltrate was an infiltration of sort of polarized infiltration of monocytes, which became these polarized macrophages that were more of a healing phenotype. So in immunology, there are different types of macrophages, and it's not, even though we sort of sometimes people like to categorize them as one thing or another, in reality, it's really more of a overlap gray zone, and macrophages tend to sort of act maybe more like one than another type of phenotype. But we found that our macrophages were expressing more of these factors that indicated that they were sort of a reparative type macrophage, and that was the essentially mechanism that we came up with for the reason why that animals that didn't have the CD39 actually did better. And what we hypothesized is that without having CD39 present, it was actually causing an increase in ATP and ADP in this extracellular space around the infarct, which actually recruited more inflammatory cells to the area. And perhaps just because of their absolute numbers, or perhaps because of the purinergic signaling, these animals were actually more induced to become these M2 type macrophages. And we showed this in some ex vivo experiments, trying to cause macrophages to become more polarized towards M2 with these extracellular purines, and that did seem to have an effect. And so after many rounds of revision and actually going through more than one journal, in fact, we actually, and that's sort of one of the teaching points from this, is that even though you look and you're like, okay, this is great, we've got this great JCI Insight paper, this actually was this manuscript actually had gone through, I think, 3 rounds of very extensive review at Nature Communications. And wasn't I disappointed when the editors changed, and then the editor that finally reviewed it after round 3 said that it didn't meet priority for publication. Fortunately, we sent it to JCI, and they transferred it to JCI Insight, and they accepted it without hardly any revision. So that was just kind of a good learning process. And so, the other thing I wanted to mention is that right around that time, so you can see my paper was published in 2017 as we were kind of going through these revisions and working towards publishing that paper. This actually was published in JCI, Alternatively Activated Macrophages Determine Repair of the Infarcted Adult Murine Heart. And this was published, I think, about 4 months or so before our paper was published. And I think, if anything, it was one of those great scientific validations where this group, although it was a slightly different model, they actually had very similar findings to what we found. And I felt very good about our results, and felt very strongly that, even though our initial findings were counter to what had been published, they were very consistent, and we didn't give up on publishing the data. And ultimately, even though it was painful to go through many rounds of revision at a high-level journal and then not get it accepted, ultimately those revisions did improve the manuscript and the quality of the science. And I think that's just kind of the way you kind of have to think about these things, including grants that don't get accepted, and just trying to think through the responses and feedback from these very smart reviewers, and try to be responsive. So that's that kind of first major manuscript that I published when I was a trainee. And then I wanted to just talk a bit about some of the work that we did also with ectonucleotidases and atherosclerosis. This work is also, again, in the context that I had a very smart colleague with me when I was on the T32 training grant, Yogan Kanthi, who, as you can see, has published this manuscript flow-dependent expression of ectonucleotide phosphohydrolase 1 and suppression of atherosclerosis. That's just another word for CD39 and atherosclerosis. And then around the same time, I was also working on CD73 and atherosclerosis. And so, the two of us were kind of working parallel benches in Dr. Pinsky's lab. Yogan is now a Lasker Scholar at the NIH and doing great things. So I'm very proud to have published with him in the past. But Yogan did some very interesting work looking at the role of CD39 and atherosclerosis, and essentially did some very interesting modeling looking at how the ectonucleotidase is expressed depending on whether or not there's shear stress, laminar stress, and showing that on sort of more healthy regions of the endothelium, the CD39 is expressed, whereas if there's turbulent flow and atherosclerosis, it tended to not be expressed. And these are some kind of cool images down here at the bottom, looking at essentially whether or not ATPase is present. So ATPase meaning the activity of CD39, and demonstrating in this serum chloride assay that that these areas where we were able to detect CD39 actually corresponded with the activity, which is very challenging to be able to do. So this is the work that we were referencing in the introduction, which I was really excited to have a cover in ATVB in 2020. That I felt like that's been so far like one of the pinnacles of my career. And then I also just wanted to point out a couple of authors on this work that were included, though they were a couple of undergrads that worked with me through the undergraduate research opportunity program at the University of Michigan. And I just wanted to, I've actually, since that publication, I've tried to do everything I can to include trainees of various sort of career phases in all of my work, because I think it is really important for people to start getting that experience even early on. And they did a lot of work on this, and they definitely deserve the authorship. So this manuscript was another one where we were kind of working in the context of another study that had showed results which seemed to make sense with what was known at that time. So this is a mouse model of atherosclerosis using an ApoE knockout mouse, which has increased lipids, and so therefore they develop more atherosclerosis. And then also this was a study that we actually did a double knockout. So the animals both had an ApoE knockout as well as a CD73 knockout. And the context for this was that there was a prior study published a couple of years before that had looked at this question. And at this time, we were actually working in parallel, but this group had actually published their work before we had. And essentially, they had showed that the lack of CD73 promoted atherogenesis. Okay, again, this is kind of like implying that the presence of this molecule was important and protective. And so I wanted to just explain that we actually did find that as time went by, that sort of pattern reversed, and that actually we found that CD73 was actually not protective in later years of an animal's life. So looking at a murine model. And this on the right-hand side was one of the ways that we explained to the reviewers why, although at first glance it seemed that our results were completely counter to what this other group had shown, you can see that if you march out these little dots, so with time, so we've got our group, Sutton, and then Bookeiser's group, their time point that they looked at, that you can see that these lines, they are kind of more actually overlapping, if anything, at early time points, and then they start to sort of go in different directions. And we see this in clinical trials. So it shouldn't be like a huge surprise to us that actually sometimes the relationships with scientific studies actually can change over time. And to me, this was, again, one of these observations that made me kind of think about the fact that a lot of times when we're doing these animal experiments, we're only looking at, 1 or 2 time points. It's very time consuming and actually uses more animals, especially when you're having to do surgeries that are sort of sacrificial for the animals, but it is important to look out at later time points, because isn't it true that especially in humans even, that a lot of times our disease state is actually happening in later life. And so, looking out to, potentially even 52 weeks in a mouse, because mice actually in captivity can live 2-3 years. So it's kind of artificial for us to be doing experiments on animals and only looking at early time points of like 8 or 12 weeks, which really represents an animal in its youth and may not represent what actually happens in later life. So we had this observation, and again, we were trying to understand like why this might happen. So we looked at a lot of different things. There were no differences in the animals' hemodynamics, their blood pressure, their serum lipids were actually very similar. Their collagen density looked very similar in the plaques. There was no difference in apoptosis. Their inflammatory cell infiltrates looked the same. There was no difference in adenosine deaminase expression. And essentially, the only thing that we could come to, which doesn't really surprise us that much, but there was a difference in the AMP to adenosine concentration in these animals as measured by high pressure liquid chromatography. And so we figured it had to do, something with adenosine signaling. And so we really worked very hard to try to figure out if there was any way that that was having an effect in terms of why there was more atherosclerosis accumulation in the animals that had CD73. And what we came to was that based off of some prior studies demonstrating that adenosine actually can prevent lipolysis, that we actually were able to demonstrate that using the plasma from these animals, that there was less glycerol production in the ApoE mice compared to the double knockouts. And that was the mechanism that we ultimately settled on, although it still felt a bit unsatisfying, and I'm still not totally sure that that's what it was, but fortunately ATBB was kind enough to accept our paper with that information. So just to summarize that scientific piece, CD73 exerted a anti-atherosclerotic influence in early time points, but with time the pattern reversed. And ultimately, some of my personal observations from this experience was just, sometimes it can be difficult to make sense of things that are counterintuitive, but ultimately, I think initially I was thinking that the other studies results were contradictory, but when I really plotted them out and looked at them, it really was eye-opening. And I just would encourage people, especially when you're looking at other studies, to really look very critically at the data in terms of even the concentrations that they're seeing, and see if it matches up with what you're seeing. And it doesn't hurt to honestly repeat things that other people have done to make sure that you can actually regenerate the data that others have said that they can do, because unfortunately there is scientific fraud out there, or people just have certain circumstances that can only work under certain very specific circumstances. And if that's the case, then maybe it is very difficult to generalize. This manuscript also kind of piqued my interest in the field of biological aging. My award that I have right now, the K award I have, is from the National Institute on Aging. And it really, kind of again showing the duration and dose of the exposure is relevant in determining the overall impact on cardiovascular aging. And this is just one other manuscript that's in press right now. And one of the reasons I wanted to highlight this was that it was a really cool experience working with he was actually working on a PhD at the University of Michigan in my mentor's lab, Dr. Pinsky, at the time, and Drew was a math major. He's kind of a fun guy. He's got math tattoos all over his arms, and so what we worked on together was doing some mathematical modeling with MATLAB, and looking at some of the previously published manuscripts in enzymatic kinetic modeling of ectonucleotidases, and essentially found some discrepancies between the data as produced scientifically and the models, and actually worked to create a model that fit the data better. Anna McGinnis and Aman Tahir are two undergrads again at the University of Michigan, where I'm really proud of our ability to work with undergrads and get them through 4 years and have them be able to graduate with a with a real scientific manuscript as first authors. So I'm really excited about that manuscript. And also it was a great experience working with an individual from a different discipline to add depth and dimension to the scientific questions that we were asking. And then just training the next generation of scientists to think very carefully about things. So I wanted to just kind of sort of phase into just talking a bit about aging research in general. I think this is something that I've been thinking a lot about as I've been talking with the other biologists of aging that I've interacted with. That there's actually very few of them who are in cardiovascular medicine. A lot of them are straight PhD, hardcore scientists, and they've been really wonderful to interact with, and I've learned a lot from them. So I think that some of the things that we do not do well in studying the biology of aging is there's oftentimes a sense that when we're talking about cardiovascular disease, we are talking about aging, and that's not exactly the case. Because I think that when we're thinking about cardiovascular disease, a lot of times we're thinking about the fact that patients actually are presenting with age. Age is one of the risk factors for the development of their disease, but they have a host of other comorbid conditions which they're being exposed to over their lifetime. And so studying the biology of aging as a discrete variable is very difficult. Unfortunately, it's difficult to do in humans. They live a long time. It's very costly. There are actually some really neat studies of human longevity, some groups that are doing research on populations that are very long-lived and very probably genetically related individuals. And that's been very cool, but ultimately it is very difficult to do. There's also, I would like to just kind of introduce this idea, if you're not familiar with it, the idea of biological versus chronological age. I know we've all, once you say it, it's kind of like, okay, yeah, of course, I know there's some people who seem to age more gracefully than others. But there's been some really neat work done by Steve Horvath and others looking at these epigenetic clocks. And what's actually kind of eerie is that we can actually look at some of our epigenetics in our genome. Those are sort of translational modifications, such as methylation or histone modifications. And that can actually detect aging in such a way that actually it predicts our longevity better than our chronological age. And it kind of also raises this question of whether or not aging actually is a modifiable risk factor. Now, not chronological age, but biological age. And are there things that we can actually do to our, for example, epigenetics that could actually sort of either turn back our epigenetic clock and improve our biological age? And I actually would argue that it is going to be possible. And there's already some preliminary work in the field of aging biology that suggests that that is possible. I also wanted to introduce this general theory of the hallmarks of aging. I call it a theory because it is a theory. This is actually the second iteration of this theory that was just published in Cell. The first iteration of this has received more than 10,000 citations. It's essentially a framework for thinking about the biology of aging and how we might study it. It includes many different things, stem cell exhaustion, as you can see, altered intracellular communication, chronic inflammation, genomic instability, telomere attrition, epigenetic alterations, as I just mentioned, loss of proteostasis, disabled macroautophagy, dysregulated nutrient sensing, mitochondrial dysfunction, and cellular senescence. Let me go back for 1 second. This particular theory is one that actually that's been augmented. They added a few in this most recent iteration that was just published. And there's been some critiques of this. So for example, this manuscript that was published was very critical of the hallmarks of aging, calling it an arbitrary list, reflecting the more fashionable and better funded areas of research. There's a bias away from medical relevance, which has now been somewhat revised in the second iteration. Neglect of age-related diseases, unsupported causes, and failure to explain how these causes actually give rise to aging, and then they're just single cause explanations. So I participated last summer in a Gordon conference of aging, which if you have if anybody in the audience is a physician scientist and ever has an opportunity to go to any of the Gordon conferences, I highly recommend them. It was probably the best scientific conference I've ever been to. They're a bit secretive and challenging to get into, but they are, I think, worthwhile. But this was a group of experts in the field of aging biology, and this is to be published work, but essentially asking people, what do you define aging as, and what causes aging? And as you can see, even amongst a group of experts in the field, this was not cohesive. And I think ultimately that still is something that the field in general kind of has to define. And so that's some context for talking a little bit here in the last hopefully 20 minutes or so about how ectonucleotidases may play a role in the development of vascular aging. And this was the topic of my K grant application, which I did also want to mention to those in the audience, anybody who's working on getting a K or thinking about doing a career development award, or even any really grant along the line, which is that I applied for a K08 initially from the NHLBI. I went through, I think, I applied for it twice, and at the end of 2 applications, and not even, I don't even know if it got reviewed, actually. It got reviewed, but I think they just, it didn't score well. I talked to the program officer, and basically what she told me was she felt that it was too risky. And I thought that was so interesting. And so, of course, I was determined to be a physician scientist. So, just talked to a lot of individuals. And I was, I'm very glad that I talked to Ray Young, who was our division chief, or is the division chief still for geriatrics and palliative medicine at the University of Michigan. And he suggested that I apply to the National Institute on Aging for my K award. And he told me about a mechanism called a Beeson Award, which is it's a K76. It's a mechanism only that the NIA has. And I was extremely grateful to have received that award. It's actually better than a K08. It's almost as much funding as R01, which is wonderful for 5 years of support. And so in the end, it was actually good that I didn't get that K08 because I got a better award. But again, it's just one of those, another experience in which initially I wasn't successful at what I was trying to do. And I just found another path to try to accomplish what I was trying to do. So, so thinking about ectonucleotidases in terms of, we were kind of explain what they do. And so I was interested in knowing whether or not ectonucleotidases might have an effect on vascular aging. There's some context for this, and that there's been some work done in a progeria model, which is the model of premature aging. There is actually a human condition called progeria. But there is a progeria type knockout mouse, and these animals essentially develop this dysfunctional protein, and they actually have been shown to have an increase in ENTPD1, which is CD39. So in these animals with increased vascular aging, there was an increase in CD39, and that was associated with an increase in inorganic phosphate relative to pyrophosphate. And as I explained earlier, inorganic phosphate actually increases mineralization. So we know, like, for example, in a dialysis patient, they have more phosphate. That leads to more vascular calcification, and the same thing happens in the animal model. And this was something that was observed. And so very elegantly, in an amazing single author manuscript published in PNAS, this author essentially showed that he could manipulate CD39 and ENPP1 to sort of preferentially shuttle extracellular ATP to pyrophosphate rather than inorganic phosphate, and actually demonstrated that he was able to reduce calcification. So with this, so there's some context for this. And then I think I also mentioned earlier this really neat work by Cynthia St. Hilaire when she was at the NIH, and looking at the CD73 or lack of CD73 was associated with increased calcification. The theory or the mechanism for this is that adenosine usually would inhibit tissue nonspecific alkaline phosphatase, which is responsible for metabolizing extracellular pyrophosphate to phosphate. And so in the absence of adenosine production, there is no inhibition of tissue nonspecific alkaline phosphatase, and you actually get an increase in phosphate production. So, and then there's a little bit of translational work demonstrating that there may be differences in ectonucleotidases such as CD39 in T cells in older individuals. And there's a little bit of data demonstrating that this is in mRNA now, that centenarians had less CD39 and CD73 compared to octogenarians. So not much is known, but a little bit was known. So some preliminary data from our lab demonstrating that we did find that in heart tissue, specifically on the endothelium, both by staining as well as by flow cytometry, that we found a decline in CD73. This is not published yet. We're still kind of working on developing this story. In a different animal model, we just were looking at this in some transcriptomic data that we had from an outbred called Het3 murine model that was aged out to 22 months and then treated plus or minus with rapamycin in late life, demonstrating also a decline in CD39 and CD73 expression in the mouse aortas. On the other hand, we have some data that's sort of negative. So from membrane protein from our wild-type black 6 mice, we did not see that same decline in aortic ectonucleotidase, although it could just be the sensitivity if I were studying it in that particular situation. And so again, this is sort of preliminary data. And we're not really sure yet, but perhaps this declining CD73 in the heart, maybe that's just appropriate and beneficial based off of our work with atherosclerosis model, suggesting that CD73 is not beneficial in late life. It could be irrelevant and not really have any clinical relevance at all, or it could be actually harmful. So we're trying to understand, how does this happen? Are there proteins which we naturally wind down? Is this passive or reactive? And that's going to be some of the work that we're going to be doing going forward. In the last few minutes, I wanted to just talk about some of the work that is actually just in press right now that we worked on, sort of the aim 3 of my K award, which was a question about cell-bound versus untethered circulating ectonucleotidases in aging. So this work essentially was trying to understand, so I told you a bit about ectonucleotidases and the fact that they're usually transmembrane proteins on cells that are circulating or on the endothelium. But there is some work that's been done, for example, by even my colleague Scott Visavati, who's at the University of Michigan, now at Ohio State, looking at microparticles expressing CD39 and CD73, or potentially they could be actually sort of cleaved from the surface of the cell and be just circulating as very small sort of membrane bound particles, or potentially acting without a membrane. So in any case, that was sort of the question that we asked. And so we were not sure if there was any relevance to this. There have been no other translational studies looking at the relationship between plasma ectonucleotidases with arterial calcification or vasodilation. And so the population that we chose to study to be able to gather all of the information that we needed was actually individuals that were coming to the nuclear stress lab at the University of Michigan and obtaining blood samples from them, and then also using all of the information that we got from their limited CT scan to be able to look at their coronary calcium, as well as the myocardial perfusion information from their PET scan. So this study enrolled 529 patients. I did this collaboratively with my colleague, Ben Murphy. And so essentially we included pretty much all individuals that were able to give us consent, that were undergoing PET imaging, and that we were able to get blood from, and just excluded a few different vulnerable groups. So we collected our blood, and then we used a REDCap database to store our individual variables. We performed the stress testing as per the clinical sort of usual process. These were individuals that were coming in for clinically indicated PET stress testing. And then we used the clinically generated coronary calcium scores when they were done, and then when they hadn't been done because it wasn't needed, we went back and did those on about 300 patients. I want to again give a shout out to Rachel Kroll, who's first author on this manuscript, and Jaha, that's in press. She was an undergrad who worked with me in the research lab at the University of Michigan, starting as a freshman in the undergraduate research opportunity program, and stuck with this project for 4 years. So again, really proud to have her be a first author. She did a lot of work with these coronary calcium scores, and we measured CD39 and CD73 by commercially available ELISAs. We actually had developed an ELISA in our lab to measure CD39, but it actually turned out to be more cost effective to just use a commercially available kit. So we ended up doing that because it had been developed in the time that it took us to develop our study. So in our study, it doesn't surprise us, but these patients were mostly male. They were in their 60s for the most part. They had a lot of comorbidities for coronary artery disease. We know that because coming for their stress tests, otherwise they wouldn't have been referred. I did just want to mention that we had a fair proportion of patients that had liver disease and were undergoing liver transplant evaluations. And then we did have patients who had prior cardiac transplant. So it was not exactly representative of a general population, and probably more representative of the population that we do PET stress testing on at the University of Michigan. So we found that there was a pretty significant range in CD39, and we actually had a lot of patients that we could not detect any CD39 in, so we ended up dichotomizing the variable for CD39 as either present or not present. We did find that CD39 was more likely to be detectable in white compared with Black study participants. We did find also that patients with liver disease and with labs that would go along with liver disease, like increased AST and ALT, actually had higher levels of CD39 compared to CD73. But somewhat to our surprise, we actually did not find any association with any of the coronary artery disease risk factor variables. And then for CD73, we also found a wide range of detectable values, and also again found that CD73 was higher in the study participants with liver disease and cirrhosis, and again with these increased AST, ALT values as well being associated. One of the things that we had been interested in looking at, because of my interest in the field of the biology of aging, was whether or not there was any difference with age. And although you kind of see a general trend with CD39 with a slight increase in the difference in the 50 to 59 age group, overall the relationship was not significant. And the same thing was true for CD73, so suggesting that at least in this population, we did not see any strong association with aging. We also looked at this relationship with calcification and did not see any association with calcification. We also looked in a sub-analysis at those individuals, the 73 participants in the study that had really no other medical problems, trying to again sort of dissect away the comorbidities from the aging question. And we did not, again, find that there was any significant association with log transformed CD73 or CD39. The key findings that we did find, though, which was I thought pretty interesting in retrospect, was that we found that for patients who had more detectable CD39 and higher levels of CD73, that they actually were more likely to have impaired stress myocardial blood flow and rest myocardial blood flow or flow reserve. And, again, we were not exactly sure what to make of this. I think if you think that CD73 is supposed to be generating adenosine, maybe you would have thought that if the patient had more circulating CD73, that they would have more adenosine, and maybe those patients would be more apt to vasodilate. But in fact, we found the opposite was true. And kind of going back to some work that my colleague Yogan Kanthi did, and some other publications, which I don't really have time to get into right this second, but had looked at other ectonucleotidase expression in atherosclerosis. Essentially, what we're hypothesizing is that these ectonucleotidases are actually being shed off of the diseased endothelium. And so therefore, in patients who have impaired ability to vasodilate, those patients, even though they have maybe increased circulating ectonucleotidase, they may not have enough ectonucleotidase locally in the microvasculature. So I also just wanted to mention that we did do a sensitivity analysis and exclude the patients with liver disease, and all of the major conclusions were unchanged. And so I don't think that that ultimately affected the results of our study. The other thing I wanted to mention is that the association with ectonucleotidases and liver disease has actually been studied. It's actually been, again, it's good scientific validation since the 1950s. There's been work in the field of ectonucleotidases in liver disease, and it's been found to affect the actual pathology and the pathophysiology and the development of liver fibrosis. So that was not a huge surprise, but again, a good scientific validation. So the limitations of the study, it's a single center study. The majority of our population was white and male. This may not be reflective of the cell-bound or endothelial expression, which we would have to investigate further. We had looked at the expression of that of the ectonucleotidase and not the activity, and it's observational and does not prove causality. So here's our summary figure from this particular study, and this will be hopefully published in the next couple of weeks. So I just wanted to kind of wrap it up by, again, mentioning that for this particular study that we found that the plasma levels of ectonucleotidases contributed to reduced stress, myocardial blood flow, and myocardial flow reserve. It also raised a consideration about the possible role of adenosine A2A receptor and the link between circulating ectonucleotidases and these PET variables. We know that the PET stresses use regadenoson, which is a adenosine A2A receptor agonist. And so, is it possible that this is just true unrelated, and perhaps this has more to do with the adenosine A2A receptor interaction with adenosine, and these ectonucleotidases are bystanders, but it is very coincidental, maybe more coincidental than maybe it's more than coincidence that these things are somewhat related, and I definitely think it requires further study, and that's something I'll be working on at Vanderbilt. The other thing I just wanted to, again, personally emphasize is that grant writing is a different skill set than manuscript writing. I learned a lot in the process of failing to get my first K award from the NHLBI in terms of that process. And as we also mentioned at the beginning, I did also receive an AHA Career Development Award and ended up declining it when I received the Beeson Award. But I think part of the reason why it scored well was because I got better at grant writing in the process. And even though I think the actual information was the same, I think just the way you present it, it's just a different skill set, and it does require additional training. And then again, not giving up on your goal or project if you think it's important just because one sponsor doesn't fund it. I also had the interesting experience, not with this specific study, but while I was in my last year at the University of Michigan, in undergoing a random internal research compliance audit for an investigator-sponsored study. And that was a very interesting process, and kind of understanding the degree to which these audits can get into details about things, where the samples are stored, what data is being stored in REDCap, who has access. I don't know if anybody in the audience has ever been the subject of one of those audits, but it's an interesting process. Unfortunately, we came through unscathed. And then also just kind of working with a knowledgeable and accessible statistician. I didn't really talk about that, but with this most recent clinical study, and I've worked with lots of statisticians with some of the outcomes research I've done, but sometimes when you're doing, your own study as your own PI, and it's a small little startup kind of study, I would say that that's probably a really important piece, is making sure you have a statistician that you can work with and feel confident in, because ultimately, with JAHA, I'd been through many reviews in the past, but this particular statistical reviewer was adding up table numbers and trying to understand the details to a degree that I'd never seen before. And ultimately, again, I felt like I felt confident in our data and our statistician, but it was a good learning process. That is the level of review that sometimes the manuscripts will go through. The last thing I just wanted to summarize again, so I think we've talked through all the learning objectives. We've talked about what ectonucleotidases do in the vasculature. We've talked about some mechanisms by which they might impact cardiovascular disease, and explained, I think I've been very transparent with you about some of the scientific challenges that I've encountered. And I'd like to share those because I think that sometimes when people come to give grand rounds, they respond, by presenting a lot of beautiful data and not kind of explaining all of the challenges that can go in behind it. And I hope that if anything, this will be uplifting to some people in the audience in terms of if you're experiencing some challenges with your scientific work, to not give up on it. This is also just this is my lab at the Vanderbilt University that I moved into in November, and it was completely empty. Some of the only things that were in there actually were garbage, so those all went in the garbage. And we're making really great progress. I have hired 3 really wonderful people to work with me in my lab, and so we've been working through all of the safety, regulatory, equipment issues, getting reagents, grants transferred, getting science done, and getting papers written. And this is my lab team, June Chen, who has moved with me from the University of Michigan. Lena George is our lab manager, and Dolly Yang is a senior scientist I recruited from the University of Pittsburgh. So I feel bad for them because she's amazing, and she's going to really help me a lot with some of the work that we're doing. So that is all I've got, and I'm going to stop there so we can have a couple questions. I really appreciate your time, and I hope I haven't bored you guys.
Thank you, Dr. Sutton. That was a great overview. I actually had 2 questions, and if anyone else has questions, please either virtually raise your hand or put it in the chat. I'll just ask them both. The first is, therapeutically, how should we or could we be utilizing CD39 or CD73 for our cardiac patients? And like what diseases, how should we administer it? Is that just not even possible? And my second question is, we have many research fellows who are interested in being proceduralists, either interventional or EP. And I was hoping you could comment a little bit on your experience with doing that, the unique challenges and advantages, and how you've kind of balanced that as you're starting your independent career.
Okay, great. So first question, I know we have very limited time, so I'll try to be brief. First question, there is not an ectonucleotidase sort of therapy at the moment. There's some work in the field of cancer biology looking at inhibitors of CD73 and specifically to try to prevent cancer cells from evading the immune system. But there's not necessarily anything right now for ectonucleotidases for cardiovascular medicine, and we're really still trying to just understand the basic biology of this. Ultimately, what I found is that the presence could be beneficial or harmful depending on the scenario. So I think that makes it a bit challenging to place exactly how this might have an effect. I think that some of our most recent data looking at the possibility of the role of ectonucleotidases with microvascular function is actually very interesting, and probably, to me, the most interesting observation that I've made so far in trying to understand, how does that impact microvascular function, which I think that's an area that we have a huge need for improved therapies in. So I think that right now we don't have anything. We can't necessarily give it for animals. We use like a potato apyrase as sort of a therapeutic sort of rescue from lack of these ectonucleotidases, but we don't have anything necessarily like that for humans. And then in my last minute or two here, I'm just talking about being in a procedural specialty and basic scientist. So I was lucky that when I was a trainee at the Boston University, I was a lab tech at Boston University. I worked with Jane Leopold, who is a basic scientist and an interventional cardiologist. She now works at the Brigham. And so I always knew that it was possible to do it. I had lots of people tell me that it wasn't a good idea. People failed, there was all this and that. To be honest, at this point right now, one of the nice things about having my K award is that I'm protected from other clinical obligations at the moment. So I'm just in the cath lab and doing research at Vanderbilt. I'm not doing any clinic or inpatient service. And, you know, there's definitely things that you give up, both on the research side in terms of time and then on the clinical side in terms of being, you know, more clinically active. So there's some things that you may have to give up, but it's very possible. And I know that we don't have much time. So if anybody is interested in that pathway, feel free to reach out to me and I can tell you more about my experience specifically.
Great. I think we have time for maybe one more question. If anyone has a burning question, please raise your hand or speak up.
I just want to say also thank you. I see a really great number of people out for a Monday morning grand rounds, and that's a brutal time for grand rounds. So I'm really impressed. Thank you for coming.
Great. Well, thank you so much for coming. Very exciting. Congratulations so far on all your progress. Appreciate it.
All right. Thank you. All right. Have a good one.
The preceding program is copyrighted by Emory University.